Hello. So 5 minutes. I was at 4 minutes and 15 seconds when I practiced, so I'll just use my extra 10 seconds there. All right. So I want to talk to you today about SeqResults. This is a system that we've been working on at NEB for a few years. I reported on it last year in its DNA form. But today, this year, I want to talk to you a little bit about the RNA work we've been doing with SeqResults, and I'll describe some more details as we go. So our goals are to capture metadata, use that metadata to identify results to compare, and compare lots of data as we're developing new sequencing reagents. So we want to improve library prep. That's the end goal here. So what kind of metadata am I talking about? I'm talking about experimental conditions like maybe it's concentration of some additive you're working on, maybe it's amount of polymerase, maybe it's a duration of an incubation step, maybe it's how many beads, how much bead volume you're using, all kinds of metadata that are unstructured. We're also talking about things like the things that we can discover, like flow cell identifiers and barcoded demultiplexing steps, and all the kinds of things that we can pull in that might be related to batch effects that we can then identify and deal with later. When I'm talking about lots of libraries, let me just share what we're doing. So we do about 20 to 30 libraries a week with our small development team, and we have to turn that analysis around fairly quickly. So we want to improve, all this is the goal of improving RNA-seq right now. That's our current sort of effort and focus. So what do I mean by improving RNA-seq? We want to have great sensitivity. We can check that box. We want a simple lab workflow. We're making progress there. We want this to be reproducible, technical replicates. We really want to see those things be very static over lots and all kinds of things. Great. We want to see that stuff. But most important is we want to reflect reality. And so what is reality? ERCCs are fairly limited, as we heard earlier in today's keynote. We want to look at those. That's my best sort of guess as to what we can do for reality. But we also want to maintain distribution of input RNA across input amounts. So let's look at some of the data. So let's look at these ERCCs. So I have expected concentration for each of these RNA, these kind of fake transcripts. And I want to see if the number of the TPM values we're getting from our data processing, we're using salmon here, correlate well. And you can see the NEB ones across the top and compared with sort of the gold standard Illumina kit on the bottom. As we change our input amount, things look pretty similar. Fairly not that correlated when you look at in linear space. People usually do this in log-log plots, and everything looks great in a log-log plot, so we try to avoid that. Yeah, so looks pretty good for the fake data, right? So for the fake transcripts. Let's see some real data. So we've picked out a whole set of transcripts that we think are likely to be real and are available and expressed in the tissue or the cell line that we're using. So high abundance looks great. Details matter. We start to see things falling apart as we get down to the really lower abundance, and that's where we want to stay focused and try to make sure that we are maintaining that transcript profile as we change the amount of input. Another aspect, coverage across the transcripts, 5:3 coverage, right? So kind of boring when you look at it in aggregate. There's one that seems to be dropping off there at the 5' end. That's the really low input Illumina one. So that's kind of interesting. Let's dig in a little bit. So this is all data that's coming out of SeqResults. So we have all this data stored. We can dynamically click and view through this data. You can see one transcript here looks really low on that 5' end, but most of them look pretty similar. So the average is really hiding something important about the data. So we want to make sure we can drill into the level of detail that's relevant. So what's going on here? And if I look at this example transcript, I just kind of, as we were preparing the talk, as we were preparing the slides for this last week, I was digging through the data and I saw, what is this? Why is there a difference here? Well, it seems one hypothesis I want to test across lots of stuff, I haven't really looked at that in detail yet, is high GC. So this goes to very high GC right here. So that seems like a worthwhile hypothesis. I want to carry that through with more experimentation and more data analysis. Right, so we want to support what we think of as best practices, looking at lots of data and lots of libraries, fast turnaround so that the bench researchers can turn their data into understanding and meaning in fairly short order. And we want to capture a lot of the metadata that could be confounding things so that we can do the sort of batch effect assessment. If you want to talk more about it, poster 22. Thanks.